The NMRS offers both primary and reference services. In this video, we will discuss the process by which the NMRS moves from receipt of a positive mycobacterial culture to generating a final report. We will cover what to send to the NMRS, the workflow within the laboratory, and the turnaround time that we aim to meet. The NMRS Reference Service takes as its starting point a positive mycobacterial culture. The majority that we now receive are in liquid media. When sending a positive liquid culture, such as that from a midget system, we require a minimum of 3 mL of culture medium in a sterile plastic universal container. Please do not send the original culture tube from your midget system. If there is any visible growth within the liquid medium, please include this in the 3 mL aliquot that you send to the NMRS. If your laboratory still uses solid media, a slope with visible mycobacterial growth is a suitable sample to send. Once a positive culture is received at the NMRS, the first step is to render the sample safe for further work. Some mycobacteria, most notably Mycobacterium tuberculosis, are hazard group 3 organisms. However, whole genome sequencing is carried out at containment level 2. To make the culture sample safe for processing at this lower containment level, an aliquot of the positive culture undergoes a heat kill process. The remainder of the positive culture is kept in a containment level 3 laboratory in case it is needed. WGS utilizes bacterial DNA. However, in a positive culture, the DNA is locked away within the nucleus of the bacterial cell. The DNA extraction process used by the NMRS utilizes a bead beating step, which breaks down these barriers. The DNA is released into a free solution and captured on magnetic beads, which hold the DNA in place while the rest of the cellular materials are washed away. At the end of this process, purified bacterial DNA has been extracted. Once purified DNA has been extracted, it has to be prepared for the sequencing process, a process known as library preparation. The NMRS currently utilizes Illumina sequencing technology, which reads DNA in sections approximately 100 bases long. It is therefore necessary to cut the long chains of DNA into short sections. A DNA restriction enzyme is used to cut the DNA into these short sections. The next step in the library preparation process is the addition of specific primers to both ends of the short DNA sections. These primers facilitate an amplification step, which increases the amount of DNA available for sequencing. They also act as anchor points during the actual sequencing process and mark the start and end of each section during the analysis of sequencing results. Once the DNA library is prepared, it is loaded onto the sequencing platform for the actual sequencing process. The NMRS uses a combination of Illumina MySeq, HiSeq, and NextSeq machines, each of which generates millions of short reads of the bacterial DNA taken from the positive culture. Once the DNA sequence data has been generated, it can be moved on to the next step in the workflow, bioinformatics analysis. The NMRS uses a bespoke software pipeline specifically designed for mycobacteria. First, it identifies all of the bacteria for which DNA sequences are present. It then selects a reference genome and maps the DNA sequences from the culture to it, generating an identification. For Mycobacterium tuberculosis, it then goes on to predict drug resistance and perform a relatedness analysis. How the bioinformatics pipeline performs each of these steps is discussed in more detail in other videos. The final step of the bioinformatics process is the generation of a report which is reviewed by both NMRS scientific and medical staff. At this stage, any further work that needs to be performed, such as phenotypic drug susceptibility testing, can be identified and arranged. The report is then validated and sent to the referring laboratory using Public Health England's eLab system. The NMRS aims to return a report for all positive cultures within 7 working days. However, if the first attempt at WGS fails, there will unfortunately be a delay as the organism is subcultured for repeat sequencing. In summary, when sending positive cultures to the NMRS, send either visible growth on solid media or at least 3 mL of liquid culture in a sterile universal container, not in the original midget container. Once received at the NMRS, positive cultures undergo a heat kill process before DNA is extracted. The extracted DNA is cut into short sections and sequenced using Illumina WGS platforms. Bioinformatic analysis is performed using a bespoke software pipeline. All results are reviewed clinically prior to the release of a final report, with a target turnaround time of 7 working days.